Aloha, my name is Orville Baldos and I'm an assistant researcher at the Department of Tropical Plant and Soil Sciences at the University of Hawaii at Manoa. And today I would like to share with you our poster on improving Peperomia Sandwichensis propagation using the application of indole-3 butyric acid and also rooting under subirrigated conditions. Peperomia sandwichensis is a potential indoor plant. This is an endemic herb that is found on the island of Kauai, Oahu, and Molokai. It is very highly ornamental because of its compact size, stem and leaf color, and slightly curved spikes. Recently, we looked at the tolerance of this species under 3 different light conditions, and we found that it can survive under office and also low light conditions for about 6 months. So if you want to take a look at that study even further, you can take a look at the abstract that was published in the 2019 ASHS conference. So now that we know that Peperomia sandwichensis can withstand low light conditions, if we want to commercialize this species as an indoor plant, then we'll need to have the propagation techniques refined even further. So in the past, we have attempted propagating whole leaf cuttings under mist conditions. However, we found it unsuccessful because of widespread rotting of the leaves. So about 5 to 15% rooting was only achieved under mist conditions. In the past, we also looked at the application of low concentrations of IBA, particularly 1000 ppm, and we did not see any improvements in rooting with these studies. So with this, we're currently looking at the use of higher levels of IBA and also rooting under indoor subirrigated conditions, because subirrigated conditions can help decrease the incidence of rotting because the leaves are not constantly wet. So in this study, we evaluated the rooting of P. sandwichensis whole leaf cuttings and also single node cuttings treated with or without 3000 ppm IBA. So for this study, we collected whole leaf cuttings that are 1.6 to 2 centimeters long with petiole intact, and also single node cuttings with one leaf intact about 1 to 1.5 centimeters long. So these were harvested from hydroponically grown stock plants. So these 2 cutting types were studied separately. So they were conducted as separate experiments looking at the application of IBA. So these cuttings were then treated with or without 3000 ppm IBA. And these were planted in pots that were filled with moistened calcined clay. So each of the pots contained about 10 cuttings each, and the pots were replicated 4 times. To keep the medium in the cuttings moist throughout the experiment, we put each pot in a 250 ml plastic container filled halfway with distilled water. So these cuttings were then allowed to root for 34 days in an air-conditioned room, so about 23 degrees Celsius on average and 57% relative humidity on average, under a white LED shop light that's turning on 12 hours daily. So this is about 6 moles per day in terms of DLI, or daily light integral. So at 34 days, we collected percent rooting. We also counted the number of roots per cutting and also measured the average total root length per cutting. Since we were only comparing 3000 ppm versus control, we then did a 2-sample t-test to see whether or not there is a statistical significance with IBA treatment. So now let's look at the results. So here you have a table. The table has 2 main columns, whole leaf cuttings on the left and then single node cuttings on the right. And then on the leftmost column, you have IBA treatment, 0 ppm IBA and 3000 ppm IBA, and then you have the p-value below. Within each of the cutting types, you have percent rooting, average number of roots, and average total root length. So let's look at percent rooting for both single node and whole leaf cuttings. So for both cutting types, you can see that there is no significant difference with IBA application. Most of the cuttings rooted, so about 90% or greater. And this is a good thing because when we were doing mist conditions, we were only getting 5 to 15%. So this is a great improvement in propagating P. sandwichensis. Now let's look at the average number of roots and also the total root length of each cutting type. And as you can see here, there is a significant effect of IBA application. When you look at things here, some of the values even actually double with IBA application. So there is definitely an effect of the 3000 ppm IBA on rooting of the cuttings. When you look at a representative sample of the experiment, you can see here the picture on the left shows you the control treatment, so 0 ppm IBA, and then the treatment with 3000 ppm IBA on the right. And as you can see a very distinct effect of the treatment. You have more roots, more profuse rooting with 3000 ppm IBA, whereas your control, there is some rooting but not as much as when you have IBA application. So in conclusion, we found that subirrigated conditions can help increase the success of propagating Peperomia sandwichensis whole leaf cuttings and also single node cuttings. And 3000 ppm IBA application can also help in increasing the number of roots and also the root length of both cuttings. So this finding can help with propagating this species and increasing the numbers so that it can be commercialized. So we would like to thank the Harold Lyon Arboretum and the State of Hawaii Department of Land and Natural Resources for providing access to the plant materials for this project. This project was also funded by the USDA Hatch project that is managed by the College of Tropical Agriculture and Human Resources. And also, this project was also partially funded by the Hawaii Department of Agriculture New Germplasm 17 grant. So if you have any questions, you can email me at obaldos@hawaii.edu. And if you want to follow us on Instagram, our handle is @UHOrnamentalHortLab. Mahalo.